Today, we are on site at the San Diego Convention Center in California at the 2018 ASH annual meeting. We'll be recapping some of the top news presented each day during the meeting, and soon we'll speak with Dr. John Pagel on data being presented across hematologic malignancies and Dr. Nubar Rajay on some intriguing multiple myeloma studies. Welcome to OncLive News Network. I'm Gina Columbus. Phase 3 findings of the Medalist trial demonstrated that luspatercept significantly reduced the need for frequent blood transfusions in 53% of patients with myelodysplastic syndromes who are anemic, required regular blood cell transfusions, and showed abnormal iron overload in red blood cell precursors prior to study enrollment. Specifically, 38% of patients who received luspatercept were able to avoid red blood cell transfusions for at least 8 weeks. In diffuse large B-cell lymphoma, results of the FLIER trial showed that 4 cycles of R-CHOP plus 2 cycles of rituximab is non-inferior to the standard 6 cycles of R-CHOP therapy in young patients with favorable prognosis disease outcome, suggesting that less chemotherapy is an option for this patient population. A total of 592 patients were randomized in this international multicenter trial, and 588 were evaluable for the final analysis. The rates of progression-free, event-free, and overall survival were comparable in both arms. Phase 3 results of the Alliance North American Intergroup Study A041202 show that ibrutinib has a progression-free survival benefit compared with standard chemoimmunotherapy in patients with chronic lymphocytic leukemia who are at least 65 years of age. However, adding rituximab to ibrutinib did not improve PFS. The median overall survival had not been reached for either treatment arm. Also in chronic lymphocytic leukemia, findings of a phase 1/2 trial demonstrated the concurrent administration of ibrutinib and the CD19 targeted chimeric antigen receptor T-cell therapy JCAR014 was well tolerated in most patients with relapsed refractory disease. The approach showed that there was also higher in vivo expansion of CD4 positive CAR T cells and lower rates of severe toxicity, specifically rates of grade 3 or higher cytokine release syndrome, compared with outcomes for a similar group of patients who received JCAR014 without the BTK inhibitor. Updated data from the Eliana trial showed that a single infusion of tisagenlecleucel lusol continues to have efficacy in pediatric and young adults with acute lymphoblastic leukemia without additional therapy. Regarding safety, adverse events were effectively and reproducibly managed. The CAR T-cell therapy continues to elicit high overall response rates and deep remissions. Moreover, the median duration of response and overall survival have not been reached with the additional 11 months of follow-up data. Also in acute lymphoblastic leukemia, results of a single institution study show that checkpoint inhibitors can effectively and safely be combined with chimeric antigen receptor T-cell therapy in pediatric patients with relapsed B-ALL. Additionally, this approach may augment CAR T-cell effect and persistence. Anti-tumor responses were specifically observed in patients with early B-cell recovery and bulky extramedullary disease. However, PD-1 inhibitors had partial but not durable effects on 4 patients with B-ALL. Moreover, no unexpected or fatal toxicities were seen. In diffuse large B-cell lymphoma, an updated analysis of the Juliet trial confirmed that tisagenlecleucel lusol produces high response rates and durable responses in a cohort of heavily pretreated adult patients with relapsed refractory DLBCL. The clinical activity was observed across all predefined subgroups, including elderly patients, those with relapsed refractory disease, and other clinical or biological subgroups expected to have a worse prognosis with available therapy. Additionally, no patients proceeded to allogeneic stem cell transplant or autologous stem cell transplant while in remission. For a full review of all of these studies, please visit OncLive.com. Here now to expand on some of the studies being presented at this year's meeting is Dr. John Pagel, who is Chief of the Hematologic Malignancies Program at Swedish Cancer Institute. Hi, Dr. Pagel. We are so excited to have you here today.
Hi, Gina. Great to be here. It's fun to be here in sunny San Diego.
Yes, absolutely. So the ASH annual meeting is upon us in 2018. So tell me, what are your top 3 abstracts this year?
Yeah, it's actually kind of hard to pick 3 because there's so many good ones. I think the biggest ones are 2 in CLL that I'll tell you about and 1 in indolent lymphomas. In CLL, you know, we've got obviously really great drugs now, and in particular we have Bruton's tyrosine kinase inhibitors or BTK inhibitors. And what we've always been doing is comparing those agents to really inferior chemotherapy regimens, and in particular, a regimen that was chlorambucil based, a regimen that probably isn't a great comparator. So now we have two randomized phase 3 trials presented here, one a late-breaking abstract and one in the plenary session, comparing ibrutinib-based regimens with real chemotherapy backbones. So the first one I would tell you about is a trial by Tate Shanafelt, that's a late-breaking abstract, that was in patients under the age of 70 that were randomized to get ibrutinib and rituximab versus fludarabine, cytoxan, and rituximab, FCR. FCR has been really the gold standard, and so this was finally a chance to compare this new novel therapy with the gold standard. And what was seen in these patients under the age of 70, that the primary endpoint, progression-free survival, was met, and significantly met. There was about a 65% reduction in the risk of relapse, progression, or death if you got ibrutinib and rituximab versus FCR. Very, very significant. And not surprisingly, because of the toxicity of the FCR, there were actually fewer deaths in the ibrutinib rituximab arm, more in the FCR arm, and that translated to an improvement in overall survival for these younger patients too. So that might have put, for younger patients, a nail in the coffin for FCR. There is one subset of patients in that trial that still might benefit from FCR. They're what we call the IGHV mutated patients. They're very favorable patients. Their FCR did suggest to be a little bit better. Maybe there was a trend, actually, I should say, to being a little bit better, but there was no difference in the arms in that small subset of patients. The other abstract that was incredibly important and will be also practice changing, I believe, is one that's presented in the plenary session by Jen Wyack from Ohio State University. And that abstract actually compared bendamustine and rituximab to two different arms of ibrutinib continued regimens. One was ibrutinib alone, and the other was ibrutinib and rituximab. And again, what was seen here was about a 60% reduction in the risk of relapse or death if you got an ibrutinib-containing regimen as opposed to the bendamustine and rituxan. It was very significant. Now, there was no difference in overall survival for these patients, and we, of course, want to follow patients for longer, but it does suggest that ibrutinib might be better for older patients, which were in that trial. They were over the age of 65. The other thing I will tell you in that trial that we learned is that adding rituximab to the ibrutinib wasn't any better than using the ibrutinib alone. So I think we learned a few things. We learned that ibrutinib is good as a single agent, maybe better than bendamustine or rituximab. But I also will caution that we learned some things in that trial too, that ibrutinib is not a free lunch, so to speak. What we learned is that there were actually more deaths in the ibrutinib-containing arms, and in fact there were more unexplained or unwitnessed deaths, suggesting that there might be unexplained deaths or sudden death from cardiac toxicity with the ibrutinib. So I think it's an important regimen, but we have to be cautious with it still, even if it's not chemotherapy. And then I guess the third abstract I would mention is the abstract by John Leonard, presented here in a trial called the Augment trial. This was a trial in indolent lymphoma, follicular lymphoma, and marginal zone lymphoma, and it randomized patients to either what we call R-squared, a very popular emerging regimen, Revlimid and rituximab, versus rituximab alone. And what was seen in that trial was that there was about a 50% reduction in the risk of relapse, progression, or death if you got R-squared as opposed to rituximab alone. And importantly, there were a large number of patients. About half of the patients in that trial had needed therapy from their initial treatment for their lymphoma within 2 years. That's a high-risk patient population. They're benefiting from this novel regimen. I think that'll also be a game changer. So I think those three abstracts are having a major, and will have a major impact in changing the care for CLL patients in the front line and in the relapse patients with indolent lymphomas.
Those are some great points you made. So now let's move over into mantle cell lymphoma. We've seen some interesting data with the BTK inhibitor acalabrutinib, which we know is already FDA approved in the second-line setting. So can you highlight maybe some of these trials with acalabrutinib plus bendamustine and rituximab, as well as long-term follow-up of acalabrutinib as monotherapy?
Yeah, acalabrutinib is what we might call a second-generation BTK inhibitor. It is different than ibrutinib in that it is a little more specific for BTK and has fewer off-target binding sites. There's pluses and minuses to that compared to ibrutinib, but acalabrutinib is approved in mantle cell lymphoma, and it's an important drug there. but it was approved in a relatively early time frame. What I mean is with a short follow-up, and now presented here by Michael Wang from MD Anderson Cancer Center is a longer follow-up of those patients, about 120, 124 patients, now with 2-year follow-up. And the good news is that the drug has held up. The responses are excellent. Overall response rate's about 80%, and the complete remission rate's about 40%. It's a very high complete remission rate. Now there's some caveats around that. These are relatively favorable or early patients in their treatment encounters, but nonetheless, high response rates, and that actually translated to very impressive durations of remission or progression-free survival. The progression-free survival is estimated at about a median of about 20 months or so. So we know acalabrutinib in the long term is a very safe drug, and we know that from that study also because we didn't have any new or emergent adverse events that came out of a longer follow-up. You also mentioned that acalabrutinib has been looked at in combination with bendamustine and rituximab. That's an abstract that's presented here by Tysel Phillips, who's from the University of Michigan, and it's a relatively small number of patients. It shows that you can do this combination, but it's got a lot of hematologic toxicity, as you would expect, combining it with the bendamustine and rituximab. And I think the jury's still out if that's a smart way to be doing this, as opposed to sequentially using these agents, perhaps one after the other.
Good. And also the agent is being looked at with more follow-up in CLL and treatment-naive patients. Can you talk about that one a little bit?
So this is also a really important abstract. It's follow-up by John Bird, looking at what we call the 001 trial. And that trial looked at about 100 patients, treatment naive, who got single agent acalabrutinib. And what that trial actually showed is that people can stay on the drug for a long time. And in fact, about 90% of patients were still on the drug at about 3 years. And there were very few progressions. Actually, out of those 99 patients, I think there were only 2 people who progressed, and there were only 3 people who came off of the drug because of an adverse event or a problem. So people can stay on the drug for a long time, and we had already shown that the response rates are excellent. They're well over 90%. And actually, if you look at survival at 3 years, 97% of patients are alive without relapse, doing well. So it's an important longer follow-up in first-line CLL as well.
Good. Now let's talk about the FLIER trial in diffuse large B-cell lymphoma. What's the data that's being presented here with that trial?
So the FLIER trial is really interesting. This is a trial that was done completely in Germany, and it was done in patients who were a little bit younger, so patients under the age of 60. And what they looked at were the most favorable large cell patients that you might be able to find. So they have an age-adjusted IPI score of 0, and they don't have bulky disease. So we know these people are going to do really well. Most all those patients will be cured with standard upfront R-CHOP chemoimmunotherapy. So then the obvious question is, well, maybe they don't need that much therapy. So what this trial did was took those very, very favorable patients and randomized them to either get R-CHOP, the standard regimen, for 6 cycles, or to only get 4 cycles of that regimen, 4 cycles of R-CHOP, plus 2 additional cycles of just the R drug, just the Rituxan. So it was a way to reduce the amount of chemotherapy. It's what was called a non-inferiority trial, and that's in fact what was found, that the 2 regimens were essentially equivalent. The abbreviated regimen was not inferior to the full regimen. So in that small patient population, this is also a game changer. This is perhaps a new standard of care. Again, a small number of patients who present that way, but important to realize we don't need that much chemotherapy.
It definitely seems encouraging.
It is, absolutely.
Now, also in diffuse large B-cell lymphoma, we have a study of selinexor. So what are your thoughts on that trial?
You know, I think the selinexor drug is one that we still need to gain a lot more experience about. You're referring to a phase 2 trial in relapsed large cell lymphoma. They're very difficult patients. And the response rate was encouraging. It was about 35% or so in there, but the durations of remission in these very, very difficult patients still is relatively limited. It's on the order of somewhere around 8 or 9 months. What the harder part is, we got to figure out the best way to actually schedule that drug, because there are some GI toxicities that can be significant and limit its use. I think we're learning very rapidly, and this trial helped, the right dose, and getting to the right schedule, and I hope this drug actually provides a significant benefit for relapsed large cell patients.
Oh, that's some great insight.
Yeah.
Now, you are also the director of the stem cell transplantation program at Swedish. So what data with stem cell transplant this year do you think kind of demonstrate the role of how transplant is evolving in hematologic malignancies?
Yeah, I would say we could have done the whole thing on transplant too. I'm going to highlight what I think is perhaps an under-recognized abstract here in refractory acute myeloid leukemia, refractory AML. Now, actually, I'm a big part of this abstract, so I'm giving myself a plug, and I probably shouldn't do that. But
okay.
This trial is a randomized phase 3 trial between a transplant regimen with a novel drug. It's a radiolabeled antibody, uses radioiodine, compared to a conventional care arm. So patients had to get, if they were randomized to that arm, some cytotoxic chemotherapy. And then if they failed that, and the primary endpoint is complete remission, if they don't achieve a complete remission, or do and it doesn't last for 6 months, which it rarely will in these very difficult patients, then they're allowed to cross over to get the radiolabeled antibody in the transplant. So while we're not presenting the actual response data here because it's too early, there's only been 50 patients accrued to the trial so far. The goal is 150. What we've learned and what we're presenting here is that the patients who get the radiolabeled antibody in the transplant all engraft on time. There's no significant increase in complications, what we call VOD or veno-occlusive disease of the liver, no increased rates of graft versus host disease, and what we also know is that if you look at the day 100 mortality rate in that radiolabeled antibody transplant, it's 0 of the approximately 25 patients in that arm so far. And then lastly, there's one other important part of that people need to know about. It's very much okay to randomize your patient to the conventional care arm because of all those patients who failed, at least 2/3 of them are able to cross over, probably even more, to get the radiolabeled antibody in the transplant. So my message there is, this is a great thing to think about if you're having a patient with refractory AML, refer them for this trial somewhere close to you, and there's about 20 centers open with this trial around the country. That abstract has potential to be a game changer.
Well, Dr. Pagel, thank you so much for sitting down with us today. It was a great time, and we really appreciate it.
Thank you, Gina.
Great. Thank you. Next up, we have Dr. Nupur Raji, who is the director of the Center for Multiple Myeloma at Massachusetts General Hospital Cancer Center and a professor of medicine at Harvard Medical School. Welcome, Dr. Raji. Thank you so much for being here today.
Thank you for having me, Gina.
So, over the last year or so, we've been hearing more and more about CAR T cell therapy in multiple myeloma. So, what updates are we hearing about with that at this year's ASH annual meeting?
So, CAR T cell therapy, obviously a very different kind of modality of treatment for patients with multiple myeloma, and we first presented some of this data now a couple years back. We did an updated presentation at ASCO, and the data keeps growing. There's so much more information at this year's ASH, and I'll highlight just a couple of those, Gina, which are kind of further along in their development. So you do have the legend data, which is being presented at this year's meeting. We'd heard about it first at ASCO more than a year and a half back, and the follow-up at that point in time was relatively short, and I think we'd seen about a 6-month follow-up there. And the difference between that and the BB2121 data that we've seen is the LCAR CD38 CAR T cell was a bispecific CAR wherein they target 2 different epitopes of the BCMA molecule, suggesting that they would have a tighter binding. And the data which is going to be presented at this year's meeting, at least the abstract looks great. It's close to 57 patients, so that's probably one of the largest single kind of center experiences with CAR T cells. There's a few differences between what we've done with BB2121 and this one specifically, and the ones which are most striking to me really are that they did not use fludarabine as their lymphodepletion treatment. They ended up using only Cytoxan. If you look at their overall response rate, really quite remarkable, close to 88%, with a very high percentage of people achieving complete responses. And again, this is the first time they're actually showing us data wherein they have a progression-free survival of about 16 months. If you look at patients who've achieved a complete response, that CR rate is close to 12, or that PFS, progression-free survival, is 22 months. I think there are subtle differences between this one and the 2121 data set, and the fact that these patients were a little less heavily pre-treated, given that it's largely a Chinese patient population and given what we know of what's available to them. So certainly they've had bortezomib and lenalidomide. As far as pomalidomide and carfilzomib is concerned, not that heavily exposed to those drugs, and certainly not seen CD38 monoclonal antibodies like daratumumab. But a slightly different population, but nonetheless, the data looks quite reassuring and very similar to the BB2121 data set. We do have others as well, Gina. The JCAR study is being presented. Again, it's a little bit early in its development, and again, using the same target, BCMA. I think they've accrued about 19 patients, and they're presenting subset of those 19 patients here, and again seeing very high response rates. A little more toxicity with the JCAR CAR-T, at least from what I could glean from the abstract. We'll wait and see what the data shows when they present it. I think it's being presented on Monday out here. But otherwise fairly manageable. CRS, neurotoxicity, and again high complete response rates. Obviously the follow-up in this patient population is a lot less, so we'll just have to wait and see. But it is an exciting time for CAR T-cell therapy for myeloma.
Good, and that was some great insight that you shared. Now looking at, there's also the phase 1 of BB21217. Can you discuss that study a little bit and the initial findings?
Sure. So the BB21217, Nina Shah, my colleague from UCSF, is presenting this tomorrow in a work session. And the difference between 2121 and 21217 is, this is kind of the next generation of CARs, so to speak. And what 21217 is trying to do is see if by manipulating these CAR T cells, can we get more persistence. And the way we're doing this here is the CARs ex vivo are being exposed to a PI3 kinase inhibitor, which is called B007, and that's why the name BB21217. The idea being, by exposing them ex vivo to this PI3 kinase inhibitor, you are going to try and get more of a memory phenotype, and you're going to try and see more persistence of these CARs. Now again, we have about I think what Nina is going to be presenting at this year's ASH is close to 12 or 13 patients, and we've seen very similar efficacy results to the BB2121 data set. So same kinds of very high response rates to the tune of 80 and 90%. And again, the follow-up in this subset is also short, but we are seeing durable responses in this. As far as toxicity is concerned, very comparable to what we've seen with 2121. CRS was certainly seen. Very little, if any, neurotoxicity. We did have one patient with neurotoxicity, but all of it was reversible and manageable.
Great. Now you're also authored on an abstract of overcoming MCL1 resistance in multiple myeloma. Can you share information on that?
Right, so MCL1 is, in my mind, a really good target for multiple myeloma. The abstract you are referring to, Gina, is a preclinical abstract, so that's coming from my lab. At this year's meeting, you are going to see data with a BCL2 inhibitor, so venetoclax, in combination with carfilzomib. And what venetoclax does is targets BCL2, BCL overexpression. Generally, when you think about multiple myeloma, it's about 20 or 30% of myeloma which is BCL2 dependent. Rest of it is MCL1 dependent. What we've studied in the lab is a specific MCL1 inhibitor. This comes from AstraZeneca, this MCL1 inhibitor, and what we've seen is one of the resistance mechanisms of preclinically not seeing responses to the MCL1 inhibitors overexpression of BCL2. So BCL2 takes over, and the data we have is combining a BCL2 inhibitor with an MCL1 inhibitor, where we see absolutely remarkable preclinical activities suggesting that this should be tested in people as well. So venetoclax in combination with an MCL1 inhibitor may in fact be a game changer for the treatment of myeloma.
Very interesting. Now, you have obviously been very involved with the BB2121 studies in multiple myeloma. So as we continue to get more data and more follow-up of this product, how do you really see it affecting the paradigm for these patients?
So, that's a great question. I think all of us are struggling with that, but anything which kind of works towards the end of disease, obviously we try and bring it earlier on in treatment, and that's exactly what we are doing with BB2121. We've actually completed accrual to the phase 2 study. That was a very similar population to the phase 1 study, and we'll see what the data looks like towards the end of next year or early 2020. And as this study is maturing, we are already planning on some of the other studies. And the other studies, and you can check those out on clinicaltrials.gov, we have Karma 2 and Karma 3. Karma 2, we are focusing more on a high-risk patient population, and we're trying to get that high-risk patient population very early relapses we're treating with the CAR T-cell approach, because that is a patient population with an unmet medical need. We know that that high-risk patient population has a very poor prognosis, and you can almost mark out the survival of these patients. So we're bringing it very early to those patients, and then again we are bringing it to the rest of the myeloma community in the Karma 3 trial, wherein we are bringing it in 1 to 3 lines of treatment. And I think this is really important, because you will at this meeting see triplets and quadruplets where the monoclonal antibodies are moving really way up front. So we are going to be using up all of this. So what we are going to do with BB2121 is studying CAR T cells in the 1 to 3 lines of treatment. So early relapse will be studied, and I do think that's going to be the future. Ultimately, I think what I would like to see with BB2121 or one of these CAR products, we don't know whether it's going to be the JCAR one or the Legend one or 21217 or BB2121, who knows, but can we kind of pitch it against an auto transplant and see whether or not we can replace an auto transplant with some of these approaches. I think all of these are just things which we will be studying in the very near future.
Great, you made some great points. Now, are there any other abstracts in multiple myeloma this year that you'd like to mention, or looking back at all this data that are being presented this year, what does this really say about how multiple myeloma treatment is really evolving?
I say this every year, but it's no joke. Every year there's more excitement, and we always feel that this year is going to be sort of a slow year for myeloma, but we don't seem to be seeing that. Now, at this year's ASH, I do think there's going to be a practice-changing trial which is going to be presented as a late breaker. This is the Maya study, which we've all been waiting for, and it builds upon the first trial. Now, the first trial really did change the standard of care for the transplant ineligible patient population, where now the majority of people are using RD, so lenalidomide dexamethasone, as sort of the standard of care for this patient population. What Thierry Francone has done very nicely in this Maya trial is now to this RD backbone added on the monoclonal antibody, so daratumumab, and the trial is 700 patient plus, comparing RD Dara versus the standard of care, which would be RD. And even the abstract you have, the PFS of the RD arm is close to 30 months, 31 months or something like that, and the PFS for the RD DARA arm is not yet reached at a very good mature median follow-up of about 28 months. So this again is going to be a game changer. We do have daratumumab already approved for myeloma in the upfront study setting, and that's in the context of the Alcyone data where it was combined with VMP. So although there was some excitement in that DARA is approved in the upfront setting, we don't really use VMP. VMP is not something, at least in the United States, which is standard of care. So the Maya trial is going to really open up the doors for the use of DARA upfront, I think. There's other data which is being presented. For the first time, we're going to see ixazomib as a maintenance option. We've always maintained, given how well tolerated ixazomib is, it is in fact the perfect maintenance trial. There was no data, but at this meeting, Panos Dimopoulos is presenting the Tourmaline data where ixazomib was compared to placebo. So I think a lot of exciting things which are being presented here and which in fact will change how we take care of our patients with myeloma.
Great. Well, Dr. Raji, thank you so much for being here today. You really shared a lot of light on some really, really interesting abstracts this year, so we appreciate it. Thank you for being here.
Thanks so much, Gina.
That's all for today. Stay tuned for tomorrow's OncLive News Network On Location, where we will sit down with Dr. Ruben Mesa of UT Health San Antonio MD Anderson Cancer Center on myeloproliferative neoplasms, Dr. C. Ola Landgren of Memorial Sloan Kettering Cancer Center on multiple myeloma, and Dr. Pinkel Desai of Weill Cornell Medicine on acute myeloleukemia. Thank you for watching OncLive News Network. I'm Gina Columbus.